Welcome, everyone. Thank you for joining us for today's webinar, A Complete Sample-to-Insight Solution for Detection and Classification of Challenging Pathogenic BRCA Mutations. It is presented by Anika Joecker, Ph.D., the Associate Director of Global Product Management for Next Generation Sequencing at QIAGEN, and Jennifer Poitras, Ph.D., the Principal Genome Scientist in Bioinformatics for Genetic Disease at QIAGEN. My name is Judy O'Rourke, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by QIAGEN. For more information about our sponsor, visit www.qiagen.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speakers will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. Joecker and Dr. Poitras. I will now turn the presentation over to them.
Thank you very much for the introduction. My name is Anika Joecker, and I will tell you a little bit about the challenges of the detection of challenging BRCA mutations. In the end of this webinar, so the second half of the webinar, I will give then the ball to my colleague, Jennifer Poitras, who will tell you a little bit more about the classification and the interpretation of those challenging pathogenic BRCA mutations, and also a case story. So what are the challenges if you want to detect variants in BRCA1 and BRCA2 by using NGS technology? The first one is that you want no gaps in both of the genes. So you want 100% read coverage of all of the exons, and maybe also the intron and exon boundaries, because you want to detect variants which play a role, for example, in splicing. The second point goes to it. If you don't have 100% read coverage, you're not able to detect all the pathogenic or actionable variants. So of course you want 100% sensitivity and at the same time a very low false positive rate. The accurate classification and interpretation is also very challenging, because after you have, for example, identified a SNP in one of the BRCA1 or BRCA2 genes, you don't know if that is pathogenic or benign, and therefore an accurate classification and interpretation is of especially interest here. So when we are looking at those challenging variants, there is a lot of bottlenecks when you try to detect them. So the first one is, for example, that you want to detect variants in Homo polymer regions. Many people are not aware of that, but BRCA1 and BRCA2 have variants in small homopolymer regions. And on this slide, you can see a stretch of A's, and there, in this case, there's a small deletion, actually in this case, not deletion, an insertion of one of the bases, which is often overseen by many other pipelines, but also by whole systems. That goes to the next point. So most of the pathogenic variants in BRCA1 and BRCA2 are small or larger deletion and insertions of all kinds of sizes. So it started with one base pair and it ends up by have the whole gene deleted. So in this case, you want to make sure to detect all of those. And that goes to the next point. So if you want to detect all kinds of sizes of deletions and insertions, then you also look at copy number variants. In the easiest case, for example, that BRCA1, for example, is deleted or amplified, but let's think about, for example, if one exon is just deleted. In this case, it will be pretty hard to detect those if you have a panel which just spans the exon regions and you don't have the breakpoints covered, which are often lying in the intron. But copy number variant detection helps you in that regard and can detect exon level copy numbers, like exon, if an exon is deleted or amplified. So the fourth point then goes after you have identified those variants. You want to interpret those. First, you want to classify them, which are the ones that are benign, which are the ones or are pathogenic. And you want not many of unknown significance because they need follow-up actions. And at the end, after you've done the classification, and also for the classification, you want to have a standardized system. So you always want to do it in the same way to also compare to each other and always give the same recommendation. You want to have a really good reporting out of those. So standardization is very important for that part. Some time ago, we asked our customers and also other people who are working in NGS, what are your pains? And what they say to us, we have a fragmented workflow and we don't know how to optimize it. And they also pointed out that they have problems with bioinformatics. So with analyzing the reads and to detect the variants and with the interpretation of the data. The analysis of the data is often connected to the fragmented workflow. So because if you have a fragmented workflow, let's say, for example, your sample extraction comes from QIAGEN, your library preparation or your panel, for example, comes from a completely different vendor, then you have the sequencing machine from someone else and the bioinformatics from the third vendor, and something goes wrong or is not really optimized. You can have, for example, the best bioinformatics on earth, but you will get bad results if the other parts of the workflow are not working well together. So the whole system needs to be optimized. And we also, people also told us they have no clue what the cost of the NGS tests are. So often, for example, maintenance costs are overseen. So someone hires, for example, a bioinformatician in their lab, and then that this person costs money is overseen for a very long time. So there was a very high fraction that couldn't tell us what they're paying per sample. But they said in general, the costs for NGS are very high and too high. I really like this testimonial from one of our customers. So he said already in 2017 that people tend to largely underestimate the true cost of running an NGS system because they're not considering the cost of an often lengthy and pricey optimization process of all of the components. So he was also one of the first customers for our GeneReader NGS system, which I would like to introduce to you. The GeneReader NGS system is not just a sequencer. So what you get is a complete solution which includes the sample extraction and it ends into the data interpretation. And all of these parts are optimized. So when we place a system into your lab, it comes with automation. So you get for the sample automation, for example, a QIAcube, or you can run it on your QIAsymphony. Also part of the library preparation is automated. You get a GeneRead QIAcube for the clone amplification and a GeneReader for the sequencing and QC Analyze for the data analysis. And what you also get is QC Interpret for the data interpretation. So as quality control, we are relying on two different parts. So one is a qubit system to check the quality of the DNA extraction, but also the library preparation, and the QIAxF system for fragment length identification. For support, there are several ways. So the good news is whatever goes wrong into the system, you can always blame us, and we are ready to help you. So we are ready with our support, and you'll get handbooks, not always on paper, but also we have a little app, so you get an iPad, which guides you through all the steps for the different assays we have on the system, from sample to insight. And what it also makes it unique, if you're running many samples, you want to exactly know which sample is at what step in the workflow. So here we have our GeneReader link system, which you can connect to your LIMS system, and where you know it's like an electronic lab book, and you know exactly where each sample is at which point of the workflow. So as I said, all the we call it panels, what you run on the system. Each of the panels are actually small sample to insight solutions, which come with their own handbook. And because of that, we promise you so that when you get the system into your lab and you get fully trained, and after installation in 30 days, you can use it in a routine way. As I said, the system is not just a sequencing box, and the things are getting pre-optimized from the sample extraction into the reporting. So therefore, the system comes with very specific assays which can be run on the system. So most of them are, actually all of them are oncology focused, except one with our custom channel, where we have also a good way, for example, to have all kinds of hereditary diseases being covered as well. So you can decide what is on the panel, which regions are targeted, and it works exactly like the other ones. So before we are putting any of those panels on the market, we are not only developing them, but we are also optimizing them with the upfront part, so with the sample extraction and the library preparation into the sequencer, and including the bioinformatics up to the data interpretation. So currently we have both 2 BRCA panels available, 2 actionable insight tumor panels of different sizes, one specific for lung, focusing on DNA infusions, and a new one for myeloid leukemia. So for today, I would like to focus on the one for BRCA. So one of those, and that's our newest edition, the BRCA Advanced panel, uses the unique molecular index technology. So the other one is, both of them are amplicon panels, but the new one, the BRCA Advanced panel, uses the unique molecular index technology, which is very fantastic to, on the first side, find out what is true and what is wrong. So for example, false positives can easily be filtered out because each of the sequencing reads, each of the biomolecules gets labeled before the amplification, and then after it gets amplified, if reads are showing the same molecular index, then you know they're coming from the same biomolecule. And if you see, for example, like this green one, where only one variant was just in one read, and the other reads coming from the same biomolecule does not share the same variant, then you know this is false positive. A true variant would appear in all of those reads. Therefore, you can easily get rid of amplification errors. And this is also very important if we think about copy number variation detection. Therefore, the new panel or the new assay can detect also copy number variation detection down to the exon level. So we tested the panel before it came to the market on different data sets. One is reference samples, and the other one is clinical samples. And here you see this was a reference sample for somatic BRCA mutations. And what is very interesting in this reference sample is that it has 3 variants in Homo polymer regions, which I said before, it was really hard to detect and often missed by many pipelines because these regions tend to many errors. So you can see here these 3, it's 2 deletions and 1 insertions. All of them are just 1 base pair long. And the third one is very interesting because it's at an allelic frequency of just 7%, so which is more rare, and therefore often also at the background level where you would not detect it. So we optimize the system to really detect also those variants. And we could show that in all samples that we detected them at 100%. So as I said, the assay can also detect copy number variation. And it can not only do that to say that BRCA1 or BRCA2 is deleted or amplified, but also down to the exon level. So how is it able to do that? So when you want to find out if something is deleted or amplified, and from a bioinformatics standpoint, you use copy number variation detection, then the copy number variation detection algorithm needs to learn how normal looks like. So you can see that here we see two, so in the middle, this we call baseline. This is normal. So normal is not one specific threshold, but really a range. And that is because you can have good or bad coverages between different sequencing runs. So you have inter-run variability. You have also changing coverage between the samples. So you have good and bad DNA. And you can have variability in parts of the components of the workflow. For example, if you change your extraction kit or if you change your library preparation kit, it will change the baseline. So the algorithm needs to learn how does normal look like, and this is pretty challenging. So what the GeneReader system with the BRCA Advanced panel did was how it tackled that in 3 different parts. The first one is that it uses unique molecular indexes, including an optimized bioinformatics pipeline to remove the amplification bias. This is very important because the copy number variation detection algorithm uses the read coverage and tries to compare if it's higher or lower than normal. The second part is that on the panel are housekeeping genes. So it tries to see on one run if it's higher or lower than the genes on the panel. And the third one, and that is what requires really a sample to insert system, is it learns, or it has learned before, how a CNV baseline looks like. So there are many samples run through the complete system. So the CNV baseline creation takes a lot of time and resources and is often underestimated for many people. So you have to run many samples through the system, and if you do it alone with an algorithm you don't know, you need to understand really well what the algorithm does. And not all the algorithms allows you actually to learn. So therefore, what we are doing, we are establishing the CNV baseline beforehand, and because we are owning the whole system, we can actually do it. So if we do CNV calling here or at your lab, it will exactly, it will have the same result because we are not changing anything of the complete workflow. A second example is BRCA germline mutation detection. So for that, this is a snapshot, not the complete verification data, but in this case, for the first parts, we used 11 blood samples and 1 reference samples. So it was partly what was clinical samples and then 1 reference sample. So we run it through the complete workflow. You can see here approximately the steps. So samples were extracted, then they were barcode labeled. The regions were amplified, so the panel does not only cover the exon regions of BRCA, but also 20 base pair into the intronic regions of each of the sites. So then the libraries were generated and run as 12-plex on the GeneReader system, and automatically then afterwards the data were analyzed with QCI Analyze. And then from there, they were uploaded to QCI Interpret. So there's a button you can click from the software and then automatically classified by using the ACMG guidelines. So we were able in all of these cases where we know the variants to detect the variants we were interested in. So we also in this case achieved a 100% sensitivity. We also didn't detect any false positives. Of specific interest here was was one variant, you can see this here, which was actually in the intronic region. So it was some base pairs into the intronic regions, and it was classified as pathogenic. So if you, for example, have a panel which just covers the exonic regions, you would have missed those variants. We also looked at the literature supporting this variant, because with splicing variants you are never sure, and there are 3 case stories, independent cases, which support this variant in the literature as pathogenic. So with that, I'd like to give the ball to my colleague, Jennifer, who will give you more insight into the interpretation and the reporting of those challenging variants and also other variants in BRCA. Thank you.
Thank you, Anika. So as Anika just mentioned, I'll be speaking about QCI Interpret. So this is the final step in the solution. So we've talked about identifying variants using the GeneReader system and QCI Analyze. So now what QCI Interpret will do is it'll tell you, which of the variants that you've identified are actually clinically relevant. So in the case of somatic alterations on the right, we automatically compute actionability using the AMP tiering system. And for germline variants, which you can see on the left, we use the ACMG scoring framework. And I'll explain throughout the talk exactly how this works. And while I'll really be focusing on classifying germline variants, especially in the context of BRCA, I do want to emphasize that QCI Interpret can classify actionability of somatic variants and alterations, as well as pathogenicity of germline variants. So for those of you who might not be aware, several years ago, the American College of Medical Genetics created this evidence-based framework to standardize variant interpretation. And you can see that there are several rules or criteria that are organized into these evidence-based categories, including functional data, for example, and segregation data. And these rules exist along a continuum from benign to pathogenic. So what the variant scientist must do is ask, which of these rules or criteria does my variant satisfy? And then those rules in aggregate will define whether or not a variant is benign or pathogenic, depending on the combination of rules that are applied. And you can read more about this in the publication cited below. So as I mentioned, one must go to basically define which of these rules the variant satisfies. The variant scientist must go to many different resources to evaluate this. So first they'll look at PubMed to see if there are variant-specific publications that are relevant to the phenotype. Additionally, they'll look in public and proprietary databases to see, has my variant been seen before, and if so, has it been seen in the phenotype that I'm interested in? They'll also look in silico algorithms as well as population frequency databases. And this presents a couple of different challenges. First is efficiency, as this manual curation process is very time-consuming and laborious. And the second is accuracy. As you can imagine, that one might miss something by looking at all these different resources. So what QIAGEN has really done is simplify this process by centralizing all this information into one resource known as the QIAGEN knowledge base. So for the past 20 years, we've had MD and PhD level scientists manually curating the literature and putting in findings in a very controlled and meticulous way. On top of that, we're bringing in third-party information, including OMIM and clinicaltrials.gov, to build this ontology such that we have a rich resource of variant to phenotype relationships so we can easily identify a causal variant in the case of disease and also classify those variants. This is an older slide, but just to give you proportionally an idea of the breadth of coverage. So on the left, you can see that the QIAGEN knowledge base contains HGMD as well as ClinVar. But on top of that, because we're curating from the literature, the QIAGEN knowledge base adds 121% more genes on top of that. And then on the right, when we look at the variant level, the QIAGEN knowledge base, because we're curating from the literature, adds 159% more variants in addition to what's in HGMD and ClinVar. So you can see the tremendous value added by curating from the literature. And just to give you an idea, I've spoken a lot about findings, but just to give you an idea of how these findings are presented. So this is a publication that we actually curated, and there are 3 findings associated with it. And you can see we list that at the top, mutant EP300 gene increases the risk, and then give you the contextual information of the disease, and then in some cases, also ethnicity information. So you've just seen how findings appear from the information that we curate, and it is these findings that are tied to the relevant rules within this scoring framework. So for this reason, we are uniquely able to automatically compute variant classifications using all 28 ACMG rules. So that significantly speeds up the classification workflow, but how accurate is our automated variant scoring? So to evaluate this, we took expert panel assessed BRCA1 and 2 variants from ClinVar and put those through QCI to compare the Enigma assessments with classifications in QCI. So I should mention that Enigma is the expert panel that is extremely dedicated to determining the clinical significance of BRCA1 and 2 variants, as well as other known or suspected hereditary breast cancer predisposition genes. So you can see up top what we've done is looked for BRCA1 and 2 variants that were expert panel assessed and put them through QCI. And in this particular example, this variant is concordant, as it was pathogenic as assessed by the expert panel, and also was pathogenic as determined by our automatically computed variant classification in QCI. So what we found, this amounted to over 6,000 BRCA1 and 2 variants, and what we found was extremely impressive. So in this graph, you can see at the very left, 81.7% of those 6,000 plus variants had the exact same classification in QCI as the expert panel. And then when we take into account differences that should not affect medical management, so just to the right, you can see likely benign versus benign, for example. When we add those together, we arrive at 99.6% concordance with respect to clinical actionability, which is very impressive. Then when we see on the right, there are these 0.43% of differences that might affect medical management. So when I started digging into those differences, I realized that the source of that discrepancy came from a couple of different places. One, the Enigma specific variant scoring criteria. So they use some criteria that's outside the ACMG framework, so we can't really control for that. Additionally, they don't consider functional studies, and we'll see how both of those are impacted in the coming slides. So first, let's take a look at the concordant variant. So for this particular variant, it was classified as pathogenic by the Enigma expert panel and also pathogenic in QCI. And you can see how this information is displayed. So in the dashboard up top, you can see that the gene is BRCA2, the transcript as well as the HGBS nomenclature, and that the phenotype is hereditary breast and or ovarian cancer. And beneath, we transparently provide you with which of those rules, which I showed you in the scoring framework in a previous slide, which of these rules were actually triggered based on information that we have in the knowledge base. And to the right of each of those rules, there's an evidence column. So if there's evidence supporting that from the literature, supporting each of those rules, we actually provide you with what evidence was used to trigger those rules. So you can see here that PS4 was actually triggered. So this is a rule that's asking, is this variant statistically enriched in cases versus controls? So since we have all these cases and controls curated from the literature and population databases, we are able to trigger this rule. And there are 24 pieces of literature that are actually supporting this. So those cases that we're curating that triggered this rule are coming from 24 publications. And I can actually access those 24 publications by clicking on the 24. And displayed below, you can see that these are all the 24 publications, as well as relevant information. Is it an individual or a family study, and other pieces of information that are provided. And you can directly link out to the abstract to any of those publications here. So QCI Interpret is not a black box. We actually provide you with the evidence used to classify these variants. So now let's for a second look at a discordant variant. So in this particular example, this variant, this BRCA1 variant, was classified as likely benign by Enigma. And you can see that the evidence that they used to support this classification was based on a probabilistic model of splicing. So when we actually put this variant into QCI, you can see that we classify it as likely pathogenic. And this is based on functional studies. As I mentioned, Enigma actually doesn't consider functional studies in their classification of variants. So we actually provide you with all the information so that you can make that judgment call. So we had 2 functional studies that actually supported a deleterious effect of this variant. And you can actually access what those 2 functional studies are by selecting that little number 2. And now you can see that we have 2 publications that actually, using a mini-gene assay, showed that introduction of this variant led to exon skipping. So we actually do have some evidence to support that this particular variant leads to a splicing defect. So you can see the power of actually having all of this evidence curated and providing what that leads to. So in summary, the BRCA panels on the GeneReader system utilize unique molecular indices, as Anika mentioned, to differentiate true variants from amplification artifacts. All panels on the GeneReader system are pre-optimized for sample extraction into the variant detection and interpretation of variants, allowing the system to detect challenging variants like CNVs down to the exon level, and also large deletions and variants in Homo polymer difficult regions. Sequencing files are automatically from there analyzed with QCI Analyze to accurately detect variants in a very easy-to-use graphical user interface. Those detected variants are then taken to QCI Interpret, which I just spoke about, to classify germline and also somatic variants using the most up-to-date knowledge. And finally, in the study I just spoke about, the QCI automated ACMG classification of over 6,000 BRCA1 and 2 variants was 99.6% concordant with Enigma expert panel variant assessments with respect to clinical actionability. And as I mentioned, the sources of discordance here were really due to nuances in the Enigma scoring framework. So I know we focused a lot of our attention on BRCA testing today, but we also have a complete solution, as Anika mentioned previously, for a variety of diagnostic applications, as you can see here, taking you all the way from sample to insight. So thank you so much for your time.
Thank you, Dr. Joecker and Dr. Poitras, for your informative presentations. I would also like to thank QIAGEN for making today's educational webcast possible. As a final reminder, our speakers will follow up with any questions you've submitted via email. That's all for now. Thank you for joining us. We'll see you next time. Goodbye.